Good morning. My name is Eric Perrault from Northwestern University, and I'm going to be presenting a talk today on asking the question, does the nervous system leverage impedance to simplify the neural control of movement? The work I'm presenting was done in collaboration with Daniel Ludwig and Mariah Whitmore. And before starting, I'd like to thank the organizers for putting together this interesting symposium. It looks like a great set of talks, and I look forward to the time that we can discuss these topics again in person. It's somewhat trivial to state, but limb impedance is essential for regulating limb stability. A nice demonstration of the importance of limb impedance for stability is this human tower created by members of the Cirque du Soleil, who are often performing in Las Vegas, where we're supposed to be meeting today. Now, because of the central role that limb impedance plays in regulating limb stability, many investigations of how humans regulate impedance have focused on tasks that involve changes to stability. One of the first studies that was done was by Etienne Bourdet's colleagues, who looked at how the nervous system can regulate the mechanics of the arm to move through unstable force fields. Let me grab my cursor here. So what Bourdet did is they trained subjects to move from start location shown here to a target location shown here under two conditions. In one case, they were holding on to a manipulator and they simply moved freely. In the second case, the manipulator was used to create a virtual environment that destabilized the posture of the limb, either by pushing it to the right if subjects deviated a little bit to the right, or pushing it hard to the left if they deviated a little bit to the left. So this is an unstable spring-like field. And what they found is that when subjects moved through this unstable field, they were able to adapt the impedance of their arm specifically to compensate for the mechanics of the environment. And that's illustrated by these ellipsoids here, which are graphical representations of the endpoint stiffness of the arm. The green ellipsoid shows the stiffness of the arm when the subjects were moving freely. And the red ellipsoid shows how the stiffness changed after subjects learned how to adapt to this environment. And you can see that the stiffness increased specifically along the direction of the instability. Now, around the same time, Denis Rancourt and Neville Hogan did a nice analysis of a different task, which is understanding how we exert forces on objects in our environment. Now, when you push against an object in the environment, the force that you're generating actually tends to destabilize the posture of the arm, as illustrated here by the use of a tool, which is one example of these force-generating tasks that can destabilize arm posture. Through a nice analysis, Rancourt and Hogan showed that there are two strategies we can use to increase the impedance and therefore increase the stability of the arm. One is to increase muscle co-contraction, and another is to change the posture of the arm so you're at a more advantageous posture position, specifically with respect to impedance and stability. Now, these two strategies were shown to be used by humans in two experimental studies that came about 10 years later. One by Sellin and colleagues out of Daniel Wolpert's lab, and one by Trumbauer et al. Out of my laboratory. What Sellin looked at is they looked at how individuals exert forces against objects of different curvatures. Objects with small curvature will tend to destabilize arm posture, whereas objects with an inverted curvature, like a cup shown here, will actually tend to stabilize the posture of the limb. And they found that indeed, as subjects interacted with these different objects, they adapted the impedance of the limb so as to maintain stability. We looked at a slightly different task. And what we did is we allowed subjects to choose their own posture as they interacted with various destabilizing force fields, similar to those used by Bourdet et al. And what we found is that subjects adapted an arm posture so as to increase stability while also minimizing muscle activity. So again, there are two different strategies we can use to stabilize the limb when exerting forces against the environment or doing other tasks that tend to destabilize arm posture. Now, as you can imagine, there are concurrent changes in how the nervous system is activating muscles in each of these different tasks. And one way we can do this is not only through volitional control, but by changing the feedback control that our nervous system uses to counter unexpected disturbances. And this is a result from a study we did back in 2010, looking again at how subjects interact with different stabilizing environments. And what we found is that the rapid feedback pathways mediated by the motor cortex adapt in how they process sensor information, such that when you're interacting with an environment that tends to destabilize arm posture in the mediolateral direction here, then the rapid feedback pathways are preferentially sensitive to perturbations in that direction. Similarly, if you change the environment so that it tends to destabilize the arm uh, in towards the body and away from the body, again, the reflexes are most sensitive to perturbations in that direction. So there are at least two different neural strategies we can employ, changes in feedback control and also changes in volitional feed-forward control to compensate for instabilities. So I think it's clear that the CNS can learn strategies for regulating limb impedance in tasks that requires a maintenance of stability. However, stability is not the primary concern in many of the tasks we perform each day. It's obviously important for some, but not all. Often we're more concerned about moving from place to place or exerting forces on objects in our environment. In these cases, the role of limb impedance is less clear. A question we have been asking recently is, can the intrinsic mechanics of our limbs simplify the neural control of movement or neural control of how we interact with objects in our environment, even when stability is not the primary concern? This is not a new question, but one that has been very challenging to answer due to the difficulties in separating the contributions from limb mechanics to those of neural control to these tasks we do each day, those that involve larger movements of the limbs and the ability to regulate the motion of those limbs with the forces we're exerting on our environment. One approach for trying to measure the mechanics of the limbs in these more functional tasks that require larger movements is to look at a variety of different motion-force relationships. One example is shown here by this study by Ferris and Farley back in 1998. What they looked at is they were interested in understanding how humans adapt the mechanics of their legs as they run across different surfaces. And they used a measure of leg stiffness that was defined as the difference between the change in the center of mass of the body and the change in the force exerted on the environment as the subjects ran across a force plate. So the change in center of mass distance in the vertical direction and the change in force was used to estimate an effective stiffness of the limb. And their results are shown here. And you can see that as surface stiffness increase, subjects decrease the stiffness of their leg, presumably so as to keep the net stiffness of the system acting on the body's center of mass relatively constant. A similar study was done by Klitas et al. Out of Andy B. Werner's group. Again, they're interested in understanding how the mechanics of the legs change as walking occurs on surfaces of different stiffnesses. Instead of looking at humans, as Ferris did, they looked at goats. And again, they looked at the slope of a moment angle curve, okay, to estimate the stiffness of the legs. They did this for each of the joints, the hip, the knee, the ankle, and the MTP joint. And the results were exactly opposite to what Ferris et al. Found. They found that as goats walk on surfaces that are stiffer, they tend to compensate by stiffening their legs. Now, there are a lot of reasons to think that the results from humans running and goats walking could be quite different, but one I want to focus on is the methods being used. So one possible reason is that it's well known that the slope of a moment angle curve represents something called quasi-stiffness, which is not a great estimate of the true mechanics of the limb. Now, let me explain this a little further. So I've already mentioned that our limbs can behave in a spring-like fashion, and this arises due to the spring-like properties of muscles. When a muscle is stretched and when a muscle is activated at a particular level and stretched, it does indeed have spring-like properties. However, our muscles are not passive structures, but rather the level of activation can change through to activation from the nervous system. That's somewhat analogous to attaching a motor to the end of our spring, right? While it's normally easy to characterize the stiffness of a spring by stretching it and measuring the force in response to displacement, if there's a motor attached to the other end and we don't know what the motor is doing, then it's not possible to characterize the mechanics of the spring simply by looking at the change in force by change in displacement, because the change in force could either be due to stretching the spring or it could be due to the actions of the motor. So the first thing we need to understand if we're going to figure out how the nervous system leverages impedance during dynamic tasks is we need a method for separating the biomechanical contributions of the limb to the forces and torques being generated from those of the nervous system. The second thing we need to understand is which type of task would the intrinsic impedance of the limb be most beneficial. So again, the properties of muscles and tendons are elastic under many conditions. And that's illustrated nicely here by this result from Joyce, Rack, and Westbury back in 1969. It's a classic series of studies on muscle mechanics. And what they showed, what you're seeing here is that changes in muscle length and changes in tension in each of the dashed curves show the length tension properties of a muscle for different levels of activation. And the solid curve showed the change in force for a given change in length of the muscle. And you can see that no matter where a muscle is in its operating range, when you stretch it, it increases its force. So that muscles behave as a positive spring, always serving to oppose any change in muscle length. Now, these spring-like properties of muscles and also the tendons attached to them have been shown to be advantageous for a number of tasks across a number of species. One study I like is another study here out of Buehner's group, who looked at hopping in wallabies. And now what you're seeing here is they found that the elastic properties of both muscles and tendons play an important role in this hopping behavior. Here you can see a motion force curve, specifically a change in length for change in force in two different muscles in the wallaby hind limb. And you can see that for a very small change in length, the muscle changes its force dramatically, showing that the muscle itself is extremely stiff during hopping. In contrast, the tendon is incredibly compliant. So as the animal hops along the ground, the muscle maintains a relatively constant length, the tendon stretches, stores energy, and releases that energy as the animal is propulsed into the air again. And the differences, so the elastic recovery of the tendon is shown here. The total work done by the tendon is on the top. In contrast, the muscle does very little here. So by leveraging the mechanics of the muscle tendon unit, wallabies are able to hop very efficiently through their environment. Now, not all tasks are similar to hopping. In not all cases, again, are the intrinsic mechanics of our muscles and limbs appropriate for assisting a task. So what we wanted to ask is, do humans leverage the impedance of the limb? And does that simplify our neural control when the demands of a task are aligned with the mechanics of the limb? So let me explain that a little further. So I consider a task to be consistent with the intrinsic musculoskeletal mechanics if the angle torque or angle moment curves are such that the effective stiffness or quasi-stiffness of the joint is required to be positive, because again, our muscles always have a positive stiffness. In contrast, tasks that are inconsistent with the intrinsic mechanics of the joint are those that require either negative quasi-stiffness or simply to require a constant force in the presence of external disturbances. In terms of trying to understand whether these different tasks are different in terms of their simplicity of neural control, we looked at 3 different metrics. We looked at how the muscle activation changes across different tasks. We looked at how difficult they are in terms of the perceptions of the subjects, and we looked at increased performance or changes in performance. So most of the data I'm going to show you today was collected at the human ankle. The subjects in all of these experiments were rigidly attached to a rotary motor, as shown here, and the motor was used to move the ankle back and forth, and also to apply small stochastic perturbations we used to estimate the impedance of the ankle. During each task, subject was asked to maintain a particular target torque about the ankle, and they were assisted in that by visual feedback on this LCD monitor. So subjects performed 3 different tasks in this experiment, and one of these was consistent with the intrinsic mechanics of the ankle. Again, the motor moved the ankle sinusoidally through a range of motion, and this sinusoidal motion was slow, having a period of about 2 hertz, or sorry, a half a hertz, period of 2 seconds. And the first task that the subject was asked to do was to generate a torque that was completely in phase with the imposed motion. So now if you plot the change in ankle versus change in the torque curve, this creates an effective stiffness, or this behavior has an effective stiffness or a quasi-stiffness that is positive, as shown here. So this is consistent with the stiffness of the ankle, which is also positive. The second task had a negative stiffness, and now the target torque was 180 degrees out of phase with the imposed motion. And this is a task that is inconsistent with the intrinsic mechanics of the ankle. And finally, the third task required the subject to maintain a steady force level of 15% maximum voluntary contraction. This should be percent NVC, not Newton meters. And the subjects had to maintain this constant torque even though the ankle was being moved. I'd like to explain now the analysis that we did in order to try to separate the influence of the intrinsic mechanics on the torque that was generated from changes in neural control. Now, as the ankle was moved through this range of motion here, this large, slow sinusoidal torque, we also imposed a small pseudo-random binary sequence on top of the perturbation, and that change in displacement, along with the corresponding change in torque, was used to perform a non-parametric system identification that allowed us to characterize the full impedance of the ankle throughout this range of motion. So we had a time-varying estimate of ankle impedance. We then took that time-varying estimate that was estimated by the small signal analysis and used it to estimate the torque due to impedance by running this large sinusoidal motion that was imposed at the ankle through our model to generate the torque due to impedance. We ran a second experiment in which the subjects had to also generate a sinusoidal torque against the motor, but in this case, the ankle was held isometrically. We measured the electromyograms from the ankle flexors, the tibialis anterior, and the ankle extensors, the medial gastrocs, the lateral gastrocs, and the soleus muscle. And we created a really simple model that looked at the net EMG. And we compared that to the torque that was generated at the ankle, and we created a static model between EMG and torque. Under these isometric conditions, the static model accounted for 98% of the variance accounted for of the torque that we measured. We now took this static model and we used it to predict the torque due to muscle activation in our movement experiments. So again, we measured the EMGs from these same 4 muscles. We ran it through our model and we generated a torque due to muscle activation. We now had estimates of 2 components of our torque: the torque due to impedance, which is due to the intrinsic mechanics of the limb, and the torque due to muscle activation, which is what the nervous system has to do to complement the mechanics of the limb or to act against them in tasks where the impedance of the limb was not consistent with the task that was being performed. And so we sum these together to get a net prediction of torque. These predictions worked quite well. You can see an example for one subject shown here. The torque that they generated in an experiment is shown in black, and the torque from our model is shown in purple here. And if we look across the 3 different tasks the subjects had to do, the task that was consistent with the intrinsic mechanics of the limb, which I call the plus stiffness task or plus K task, the task that was opposite to that required the behavioral stiffness of the ankle to be negative, or a task in which the subjects maintain a constant torque. The model errors in all of these were similar and less than about 4% maximum voluntary contraction. So now let's look at the results. How did the subjects complete each of these tasks? I'm going to walk you through results showing you examples from individual subjects on the left here in these time traces, and in the polar plots, I'm going to summarize the results across all subjects. So on top, we're showing the motion of the ankle that was imposed by the motor. On the bottom here, we're showing the torque generated by the subject in a single trial. And you can see this is a torque from a task that was aligned with the mechanics of the limb, since the torque was completely generated by the subject is in phase with the ankle motions. And this is represented here on the right in terms of the polar plot. Again, we're going to reference everything to the ankle motions, which I'm going to reference as 0 here. Each of the dots down below, these are the results for the torque. Each of the dots show the results from one subject, and the vector shows the average across all subjects. And you can see that the subjects were able to generate a torque that was pretty well matched to the target. Now we're going to look at the component of that torque that was generated from the impedance of the ankle. And you can see that the impedance contribution to the net torque was again in phase with the motion, as you would expect. And it had a magnitude that was fairly close to the target, but not all the way there. The difference was made up by muscle activation. So this red curve here shows the muscle activation torque from a single subject. And again, the group results are on the right. And you can see that if you add the red and the blue, you get close to the black here. You'll notice that these don't add exactly. And that's again because the models we generated were not fit to the experimental data that we're using them to predict. How about in the case where the subjects had to maintain a constant torque level? Okay, so you can see again, the ankle is moved through the same sinusoidal trajectory. The black trace shows what the subject was able to achieve. They were able to hold the ankle torque relatively constant at about 15% maximum voluntary contraction. Again, the torque due to impedance, shown here in blue, is in phase with the motion. And now what the nervous system has to do is has to generate a muscle activation torque that opposes that to cancel to get this net torque shown here. Again, if we look at the group results, we can see that the subjects were able to maintain the constant torque close to zero, as shown by these black dots. The torque due to impedance was again in phase with the perturbation, with the imposed sinusoidal perturbation, excuse me, and the torque due to muscle activation was opposed to that, countering the intrinsic mechanics of the limb. And finally, you can see that in the case where the behavior required the subjects produce a quasi-stiffness or an effective stiffness at the ankle that was negative, again, we can see that the intrinsic stiffness was opposed to what the subjects were trying to do. So the muscle activation torque had to be large in the opposite direction to what the stiffness was doing. You can see that in the time trace here. Again, the subjects were able to create a torque that was 180 degrees out of phase with an imposed motion. The intrinsic stiffness or the intrinsic torque was in phase, and then the muscle activation torque was large and opposed to counter the effects of the mechanics of the joint. If we quantify these results across subjects, we can take a look at the magnitude of the muscle activation torque. So this is a measure again of what the nervous system had to do in each of these tasks. We're going to look at both the mean torque level and also the standard deviation of the torque. And the standard deviation you can think of as a measure of how hard the nervous system has to act by changing muscle activation throughout the course of these trials. And you can see that in the plus K task, which was aligned with the intrinsic mechanics of the joint, the nervous system had to act less. Muscle activation was lower, both in terms of the mean activation or the mean torque generated by muscle activation, and also the standard deviation of the torque that was generated. In contrast, in the case where the subjects had to generate a behavior that had an effective negative stiffness of the joint, the nervous system had to work hard, generating both a large mean torque and a large standard deviation of torque. And finally, as you'd expect, if we look at the contributions to the torque from impedance, they were almost exactly the opposite. So in the case where impedance was beneficial, it had the largest contribution to the torque. In the case where impedance was most detrimental, it had the least contribution to the overall torque, both again in terms of mean and the similar trends, although less statistically significant in terms of the standard deviation of the torque. Another way we tried to determine how difficult these tasks were, or how much of a challenge they were to the nervous system, was simply by asking the subjects. We asked them to rate each of these tasks in terms of the level of the difficulty from 1 to 5, with 1 being easy and 5 being hard. And the results are shown here. You can see that subjects found that the task that was aligned with the mechanics of the joint was by far the easiest of the 3 tasks. The most difficult task was actually that in which they were asked to maintain a constant torque, which had an effective stiffness or behavioral stiffness of 0. Finally, these results are not due to differences in performance because subjects were able to track the target torque with equal amounts of accuracy in each of the 3 tasks. So the error in the tracking was about 3 to 4% maximum voluntary contraction, whether the task was aligned with the intrinsic mechanics of the joint or even when it was not aligned with those mechanics. And I mentioned before that we measured the impedance of the limb. And so generally in terms of musculoskeletal impedance, it's often been shown that the impedance of limbs and joints can be characterized by a system with inertial, viscous, and elastic components. And we found that the elastic component was the main contributor to the torque we measured in these experiments across all tasks. An example in one task is shown here, where you can see the torque predicted by the stiffness of the joint, the viscosity of the joint, and the inertia of the joint over the time course of part of a trial. And again, the stiffness component dominates. And this result was consistent across all subjects and across all tasks. So again, the yellow trace here shows the component of the torque, there's standard deviation of torque predicted by stiffness, and it dominates in all tasks. And so this kind of justifies in a post hoc fashion the protocols that we use, where we generated tasks that have different behavioral stiffnesses, ignoring viscosity and inertia. And it turns out that the stiffness was actually the dominant component of the mechanics during these different experiments. So we found that tasks that require users to generate torque angle relationships that have an effective positive stiffness are easier to perform and require less muscle activity than tasks that have different torque angle relationships. What I'd like to talk about now is how do we go about performing tasks that are inconsistent with the intrinsic mechanics of the joints? There are many ways that the nervous system can compensate for the intrinsic mechanics of the joint when those mechanics don't help us. We addressed this question a few years ago by asking subjects to hold a constant force while being continuously perturbed. These experiments were conducted at the human elbow, but then they're many ways very similar to the experiments I just showed, particularly when subjects was asked to maintain a constant torque about the ankle in the presence of those large sinusoidal perturbations. We compared the performance in this torque control task at the elbow with the torque generated at the elbow when the subjects were asked simply not to respond to the perturbation, which I call a do not intervene task, which is indicated by this DNI acronym here. And you can see that subjects were able to reduce the torque when instructed to do so fairly well. So you can see these large variations in torque in the do not intervene task that disappear when the subject is doing the torque control task. There's still a lot of high frequency response to the perturbation, since the nervous system can't respond extremely rapidly to the small scale perturbations that were applied, but they can compensate at least for these slower changes in torque. How can they do this? They can only do this by reducing the impedance of the elbow. And so the impedance of the elbow that we estimated is shown here in the frequency domain. These are average results across all subjects and the two different tasks. Again, the do not intervene task, which is shown in blue, which has a relatively high impedance, particularly at low frequencies where stiffness is known to dominate, compared to the torque control task where the subjects were able to lower their impedance at low frequencies, below frequencies of about 1 hertz or so. And the question we wanted to know, particularly relevant to the talk I'm giving today, is how is this accomplished? How could the nervous system lower impedance so as to counter the intrinsic stiffness of the elbow? And we found that it did this through changes in feedback control of muscle activity. We examined this by looking at how muscle activity changes in response to the perturbations that were applied at the elbow. So this is an impulse response function showing changes in muscle activity of an elbow flexor, the brachioradialis, in response to elbow perturbations or elbow accelerations. And we can see, first of all, that if the perturbation is applied here at time 0, you can see a large change in the magnitude of muscle activity that occurs at a short latency of about 30 milliseconds. This is consistent with the short latency stretch reflex. And you can see there's a rapid increase in muscle activity shown in both the do not intervene task and in the torque control task, showing that these spinal reflexes don't change their sensitivity from one task to the other. However, what happens soon after the EMG begins to recover from the stretch reflex is that it differs. It begins to diverge such that we have a maintained activity level in the do not intervene task, but this activity returns towards 0 and actually begins to assist the perturbation in the context of the torque control task. So subjects are able to, while the stretch reflex opposes perturbation and enhances the spring-like characteristics of muscle, we found that in the do not intervene, torque control task, subjects are able to counter the stretch reflex and begin to assist perturbations at a time scale of about 150 or so milliseconds. We wanted to see if this change in feedback control had an impact on the mechanics. So we plotted the change in the impulse response functions against the change in impedance that we measured, and we found that there was a strong correlation between the two. So this provides one mechanism for overcoming the intrinsic mechanics of our limbs when they're not beneficial to the task being performed. showing that the nervous system can either benefit from the intrinsic mechanics of the limbs and use those to simplify neural control, or that it has strategies for adapting neural control in tasks where the intrinsic mechanics of the limbs are not aligned with what we're trying to do. So in conclusion, I've shown that tasks that are consistent with the intrinsic mechanics of our limbs require less modulation of muscle activity. I've also tried to show that these same tasks are perceived as being easier to perform by the subjects. And finally, that we have rapid feedback pathways that allow us to compensate for the intrinsic mechanics of our limbs when those mechanics are inconsistent with the task demands. I'd like to end by thanking you for your time and also thanking the members of our laboratory, particularly those who contributed to this study. Daniel Ludwig, a research professor in my group, who did much of the analysis and came up with many of the ideas of this study, and Mariah Whitmore collected all of these data as part of her PhD. Thank you.